Okay, good morning to everyone. Welcome at today's talk. There might still be some people waiting to log into the room, but in the meantime, I'm going to go ahead and introduce Nicolaus. For those of you that don't know me, I'm Reinier Dennis. I'm at the Center for Water Sciences and Management at the Northwest University. So just a bit of background on Nicolaus. He completed his chemical, and then after that we appointed him as a lecturer, and he worked for us for nearly 4 years at the NWU, and then after that he left for Luxembourg to go and do his PhD. So he's busy finishing up with his PhD this year, and he did it at the Luxembourg Institute for Research and Technology, and he agreed today to share with us some of the research that he's been working on the last couple of years. So thank you for that, Nicolaus, and I think Ilanda will allow you to share your screen, and then we can continue. Oh, before I forget, for those of you, I think we all know the drill. You're welcome to post comments or questions in the chat box, but there'll also be time for questions afterwards. Yes, so thank you, Nicolaus.
Thanks, Reinier. Thank you for the introduction. So let me just share my screen. Share. Okay, perfect. Hi guys. Thanks for joining. It's quite a big turnout. I didn't expect so many people to come and look or see what I have to do say, but hopefully it will be interesting to you. So the title of the talk today is a stochastic modeling approach of PHREEQC using multiple natural traces to track solid geochemical evolution along a flow line in a forested headwater catchment. So just to give a bit of background, I'm moved away a bit from mining geochemistry, and I'm working now more in so the PhD is sort of a bridge between hydrology as it is practiced in Europe. It's a bit different than what you probably are used to in South Africa. It's much more focused on travel times. It's a big field in terms of trying to predict floods. So really into the stable isotope geochemistry and so on. But my side is a bit more looking at geogenic solutes, so calcium and magnesium, stuff that might be used as traces that are coming from weathering of slates, especially in our catchment. So how the presentation will go is I will give a brief introduction about my topic and the data we work with. Then I will go a bit into the multivariate analysis. We did a substantial amount of statistical analysis and a bit of machine learning techniques to sort of untangle the whole process, or all the processes might be taking place. And then I'll just show how the model is working, and then I'll move over to what we did in terms of geochemical modeling to see if the behaviors that we observed through the multivariate analysis is actual fact things that's happening. Or so in our catchment, so it's not in the sandstones, it's Darwinian slates. Sorry, I'm not a geologist, so I might be mispronouncing some of this stuff. I apologize for that, but the basic things are it's just slates. But what makes this catchment really interesting is that the soils are not autogenic, meaning that they were deposited there via glacier movement and so on. So in normal weathering models, you would expect that the soils would form from the bedrock and that they will be depleted in weathered material. But this is in our case not what is happening. We still have some primary minerals in the soil. So you have weathering in both the soil and the bedrock. So your sort of weathering front that is moving down is in various stages of weathering. So in terms of what is happening in flow generation, so the whole process or the whole PhD project is centered around trying to figure out how to predict the discharge chemistry, so meaning the little stream in the catchment. And a lot of isotope work has been done and they are fairly confident with the type of flow lines that you can observe in terms of stream flow generation, because we have what is often seen in headwater catchment is a double peak flood response. So in terms of rainfall, you have an initial hydrograph peak, and then it subsides, and then the secondary peak, or a much larger and continuous peak, arrives. So it is mobilization of older and newer waters that is stored in the catchment. So to sort of just simplify it in a schematic, what you can see here is a transect of the hill slope, and then you have a highly fractured slate bedrock, and the flow line that moves through that is flow line D. And then you have this sort of weathered, really weathered, where you still have pieces of rock and soil and so on, or mineralized soil mixture. So there you have a flow line called B. This is the one that is in close proximity to the stream. And as the system wets up and you get sort of the saturation wedge that forms up the slope, you start getting a flow line C. So this is a mixture of what is rapidly infiltrating through the highly porous soils, and then mixing on this sort of contact between the weathered and unweathered zone, and then it flows to the stream. And then we have quite some substantial influences of the riparian zone around the stream. So this also alters the chemistry. So in terms of the data set, the list has been monitoring this experimental catchment for nearly 10 years. So we have bi-weekly chemical analysis of around 13 wells, but it now the monitoring grid has been extended to 27 wells. And just to give you an idea of the magnitude of monitoring, the catchment's only 14, so no, I apologize, 42 hectares. It's really small. So we have a lot of wells, and basically all of the wells that I studied are on a transect of the stream. So I could really track the flow of water as it recharges on the plateau and on the slope, and how it discharges at the stream. So I have about 5,800 odd observations with 40 parameters. So it's major cations and anions, and also ICP data, and a lot of soil solution data as well. So if we look at the Piper diagram, you can already see there's some interesting trends. So this sort of light green color is a throughfall, because the catchment is covered by a mixture of vegetation, so spruce and Douglas fir. So this vegetation have an influence on what is input in terms of chemistry, but also we monitor dust deposition, and obviously the pure rainfall as well. So the pure rainfall is the blue cluster, and then this sort of light brown color is the soil water composition, and the clusters that you see here are all groundwater clusters. Right, so already you can see there's sort of a trend. So you move from the freshly recharged water through the soil. So it moves from a potassium sodium chloride dominated zone. It goes to more of a calcium magnesium sulfate type water, and as it moves to the deeper part of the water, the relative contribution of sulfate decreases, and the relative contribution of alkalinity increases. So already we sort of can make a gut call and say, well, perhaps there's some pyrite oxidation, because the soil solution is relatively low, around 4.5 the pH, and as you move deeper, it gets buffered and goes to 7.5 more or less on average. So this would have been the initial guess of what is happening in terms of chemistry. It's sort of just a titration where you have acidic soil water, and it gets buffered by some base. So then what we did is, because just to quickly go back, the problem is our wells intercept various flow lines, and because you have a really high variance in terms of vertical conductivity due to the soil being deposited there and it's not weathered, you have, if the system is wet, you have really high lateral conductivity. And as it dries out, obviously the conductivity decreases. So if you were to sample in the wet season, you more or less will only be drawing in water from the shallower flow lines. And as you obviously the system dries out a bit, you start getting the contributions from deeper flow lines, or you can see the signal at least. So what we did is we just do a simple hierarchical clustering. So you can see the dendrogram on the left-hand side. And initially we cut the tree at around 6 main clusters. And we just did an assessment to see the frequency of occurrence of these clusters in our various wells that we were monitoring, and see if it would make sense. And indeed, it really predicted it quite well. So this blue is more or less what is observed in the stream, but you can see it in most of the shallow wells, meaning that what we expected, that most of the stream flow is coming from the shallower sections of our catchment, and this is broad. It can just be attributed to very high lateral connectivity versus quite low vertical connectivity. And then but what we wanted to do is to go a bit more in depth to see if we can sort of explain the clusters. And we moved the Finnan line one step down, so cutting the tree to 13 clusters. So what I'll show you then is we classified the samples, or just labeled them, and that then we did a principal component analysis. So principal component analysis is a multivariate statistical analysis where you aim to just explain the variance in the data in terms of loading vectors. So to see how the parameters that we measure can explain the variance that we see in the data, or why the clusters are separating, right? So this polygons that you see here is the initial big clusters, 6 clusters that we made. So the dark one is the sort of deeper water that we observed, and this purple cluster I'll get to in a minute, and then this red cluster and the blue cluster is the two sort of chemistries we observe in the stream. So the red cluster is base flow, so when it is dry, and the blue cluster is when it is completely wet. So and then this green cluster is on the plateau. So just to quickly give you just a bit of background, our catchment has 2 plateau or 2 hill slopes. So the one is what we refer to as the primary hill slope. So this is the largest part of the catchment, but then we have a smaller secondary hill slope. And the secondary hill slope, the soil layer on top of it, it's homogeneous, but on the primary plateau, we have quite a significant weathering front. But what is interesting is that this weathering front has a cutoff in terms of elevation. So the base of the weathering front you can observe on the hill slope. So below this on the hill slope, you have unweathered rock. So this interplay between this weathered zone where you have a bit more high conductivity and chemistry, the chemistry also is a bit different, and how these units interact with each other in terms of hydrological state is then really what this PhD is about. But I will not go into too much detail about that, because I can keep you here the whole day, and that is not super interesting for most. But I'll go into more the chemistry, because I need to sort of explain this just to show how we did the modeling. So just to get back to the PCA, so what we could immediately see is that most of the variance on principal component 1, which explains 48% of the variance in the data, is just calcium and magnesium. And then also to a lesser extent alkalinity and silica. Then on principal component 2, you have sulfate, and then you have sodium and potassium, I will explain in a moment, and chloride, and also nitrate on the secondary of the PCA2. And then if we compare PC3 with PC2, you can also see that there you again see calcium and magnesium. And then, in this case, what is interesting to note is that sulfate and alkalinity sort of have an inverse influence on the the variance, and this I will explain in a moment. So if we then again look at the Piper diagram, but we sort of take away the dots and we just look at clusters, we can look at a ternary diagram. So we can discuss ternary diagram 1 first, which is the cations. The cations. So what is interesting here is this gray area here is 500 soil water data points. So this forms this really distinct area. And then what you see here in the sort of green color is the through-fall influence, and then the blue contour lines is the rainfall. So these three components make up most of the shallow groundwater. Because we have a lot of preferential recharge in terms of a lot of rooting. The trees' roots we found carry a lot of water to the deeper section, so they sort of bypass the organic horizons of the soil and go directly into the mineralized soils. So, but then the two shallow clusters, this is what is interesting that we observed, and this we also observed in a stream, but we could not explain sort of the difference between the two, is that they have more or less the same variance in terms of calcium and magnesium, but a distinct difference in terms of sodium concentration compared to calcium magnesium. And this is solely because of ion exchange. So what happens there is there's sort of light color cluster. It is in when it is dry, you have rapid infiltration and you have vertical flow through the soil. So not a lot of ion exchange takes place. But in the wetter, when the system wets up, you have lateral flow through this horizon and you have a lot more ion exchange. So then we can already distinguish these two clusters from each other just in terms of looking at ion exchange. And then looking at the anions, we could see that again, this is the soil water here. And this is just the clusters that we observed through the hierarchical clustering. There's two clusters here, this sort of pink and black is the deepest clusters. And as you can see, most of the groundwater, as it moves deeper, it sort of converges to the deepest point. And the blue that you see in the background is all the surface water samples. So it's about, in our data set, around 3,000 surface water samples that can make this this area here. So you can see a lot of the time the surface water is just a linear combination of this clusters that we observe, but we can observe end members in the system, and this is what we're working towards. So explaining what is happening and why this trends are occurring is what the geochemical modeling is all about. So I'll discuss this a bit. So in the database, there was around 3,800 samples that at full all the data, so I could model those. So in PHREEQC, I didn't manually input everything, which you can do it in Python on R. So I ran the data and to see what how the saturation indices of the minerals look like. And what we could see is more or less what we already expected, is that most of the anions are controlled mostly by carbonate minerals. So you can see in the deeper clusters here, for instance, 1.3, 1.2 and 1.1, you have a relatively high saturation index for dolomite and calcite. And then also for gypsum, but gypsum is throughout the system. I did not show the D and XRF data or results that we found, but we more or less saw a lot of this type of sulfate minerals in the system. So anyway, when you plot the raw data, the calcium plus magnesium mill equivalents against the total alkalinity plus sulfate, you can more or less explain most of the chemistry in the system, except for two sort of special cases, and this is what's happening in the riparian zone, and I will discuss this a bit later. So this heat map really gave us a good insight into what minerals to consider in our model. So we just again check if the clusters make sense. So this is a water level versus a scaled a Z transformed calcium magnesium ratio. So meaning the mean is 0 and the standard deviation is unity, right? So it's just the scaling so that we can see the variance between points. And this you plot against the water level, and you can actually see it's quite interesting. So this is on the plateau. GW1 is a shallow well. It's about 2.5 meters deep, and just next to it, there's another well that is 7 meters deep. And what you can see is that when the shallow stuff is wet, you get a lot, or you get this green cluster, and as it dries out, you sort of get this purple cluster, and if it dries out more, you start having the dominant deeper groundwater that's really in the dense slates. What is also interesting to note is this sort of validated our hypothesis that we could see preferential flow paths in terms of chemistry. So when GW1 is really dry, so what you're really just pumping out is just seepage from the top. You can sort of you see if there's rainfall events, you see the bypass. So the water is not the recharge is not chemically altered by the soils. It sort of just bypasses and gets into the deeper part of the aquifer, or relatively deep, in 2 meters. So on the slope, this is now below the weathering front, so below the sort of elevation where the weathering front stops. You can see you have a bit of different chemistry. So here you have a very you have the slope, so there's just really the soil and a bit of weathering on the bedrock. So when the system is really wet, you see this lateral flow line cluster that we observed also in the ternary diagram. And when it dries out, you start seeing that you have vertical infiltration through the soils, and changes the chemistry, right? And if you move in deeper, you start seeing the water that really provides the base flow to the stream. And then this purple cluster that I showed you, that mostly is formed on the plateau, you also observe this on the yield slope. So what happens is that the yields on the plateau, you have sort of like a quasi perched aquifer situation. So you have this weathered material that's sitting on top of sort of a semi-leaky or fractured slate that is quite dense. So if the water level goes up to a certain elevation, and it exceeds the line where the weathering front stops on the slope, this water spills over onto the slope, and it runs, and it's practically chemically not altered at all. So it just runs on this contact between the soils and the bedrock, and it gets mixed with these two inputs. So the shallow soil waters and the sort of vertically infiltrating soil waters, and this we could also prove through mixing models. So because this you don't see in the stream, but we could see that this cluster here could not be fully explained by just mixing these two. We had to add more sulfate, and this is produced on the slope, or on the plateau, sorry. And then just near stream chemistry. So this is quite close to the stream. On the outlet, you can see sort of all the flow lines converging. So you have this the shallow flow line, and then you have this mixed flow line that is comprised of the purple cluster and the brown cluster. So most of this goes into the stream, and then you have a deeper flow line that is formed on the slope. So it's not completely the same as the deepest flow line that I showed in the initial schematic, flow line D. It's a bit younger, but still flows through the denser stuff. So this more or less validated that our classifier picked up or really did a good job at separating the water signals that we observed in the sampling, and then we could start trying to model with this clusters, right? So just this is just interesting behavior from the secondary plateau, because there's such a shallow weathered front, and it's highly fractured. Most of the And also it has a different type of vegetation that we see has a lot higher input of nitrates and chloride. So you can definitely see that it's chemically really not altered. It's just recharged, runs through the fractures, and is discharged into the stream. So just to show you quickly, what we now did is we sort of, every time we had a sample, we plotted this on the daily discharge of the four stream flow gauges that we had. So, and then you can actually see it works quite well. So when you have high flow, it's just the shallow stuff. This little point here is what I'm currently working on. This is the chemical alteration in the riparian zone. So if you have dry conditions and you have a small rainfall event, the first peak is usually just water that is stored in the riparian zone that was chemically altered a bit. It gets pushed into the stream, and then you see some of the deeper flow lines when it is really dry. Just to also give you a bit of background, our catchment the stream almost dries out completely in summer. This is due to the fact that evaporative or evapotranspiration plays a large role in the water dynamics of the system. So you can almost have a completely dry system in summer where there's almost no water, and then in winter it fills up again. So then SW2, we saw more or less the same sequence. So this again validated for us that our clustering method really worked well. And so the next step is now to really see if the clusters we observe, are they true end members in the system, or are they linear combinations, meaning that are they mixtures between clusters. Because at the end of the day, we aim to do what is called a stream flow separation, and usually it's done through stable isotopes, but you can also do it through chemistry, but you have to make the assumption really that you have a conservative tracer, which is actually not the case for most solutes or natural traces in a system. So you have to separate the mixing variance from the chemical variance, right? So this is the aim then, is to account for the chemical variance, and then what we're left with is mixing variance, and then we can start understanding what generates stream flow in the system. So this is the last one, SW3. It is situated really close to big riparian zone, so you can always see in dry periods the influence of the riparian zone, but for the most part is dominated by shallow flow lines in high flow or wet conditions. The last one is SW4, same basic story. So we are fairly confident with this results. So we will go now, to the modeling. And so we hypothesized, or we said, well, okay, there's a lot of DOC in the system. So, and potentially we didn't pick up any pyrite in the mineralogical study. So we could argue that perhaps most of the pyrite that we're seeing, or it could be most of the sulfate might be coming from pyrite. The pyrite can be secondary pyrite, meaning that we have dust deposition and vegetation cycling. This inputs sulfates. The sulfates can be reduced in summer to pyrite. It gets oxidized, and so it's a sourcing situation. Or it can just be stored as calcite, because this is also, maybe there was pyrite because the weathering front on the plateau is about 20 meters deep, and it was stored as calcite, and it just sort of leaches out. Because what we saw in the throughfall is it can potentially account for the sulfate dynamics that we see, but on average the mean sulfate value is relatively low, about 2.5 milligrams per liter, and the highest values that we observe in the deeper flow lines are about 14 milligrams per liter. So there has to be some source, or at least some temporary storage of sulfates in our system. And then we also had to account for the increase in alkalinity. So basically we could state that, okay, so the primary proton donor in our system can be pyrite, or it could be just the oxidation of DOC, dissolved organic carbon. So to do this, we can do a virtual experiment, right? So we can say, okay, say there's no pyrite in the system and all the sulfates are stored as calcite, gypsum, and we know that in the system we have calcite and gypsum, and you can have complexation. The dissolution of calcite can also drive the dissolution of gypsum. have DOC oxidizing, it forms carbonic acid, so it's just soil respiration. So we can test this in PHREEQC, right? So we can start by just saying, okay, we take gypsum, calcite, and there's also magnesium. So the question is really, is it dolomite or is it magnesium calcite? We don't know, and it's quite difficult to model magnesium calcite for this. So for this exercise, we'll just take a dolomite as a proxy. So a mixture of calcium and dolomite would probably explain our calcium magnesium dynamics. And we add some gypsum, and then we sequentially add DOC. So in this case, DOC for us is just acetate, and we oxidize that. We add 0.1 moles of 0.01 moles of acetate, and we see how the pH changes, right? So this is, if you visualize, it's just a little batch exercise. So this more or less explained the deeper flow lines actually quite well, just DOC, because we see a lot of DOC leaking into the deeper stuff through preferential flow paths. So theoretically, if you have a homogeneous porous media, your DOC will be oxidized way before the time the water arrives or recharges at the deeper part. So you have to have preferential flow paths for that to get the DOC down there. And this more or less explained what we saw. So PCO2 here is the partial pressure of CO2. So for those of you who might not know how this works, when you have DOC that is oxidized, is a combustion reaction. So you have CO2 that forms as a product, and CO2 as a gas then dissolves into the water forming carbonic acid, and the amount of carbonic acid it forms is a function of the partial pressure of CO2 and also the temperature. So if you look at Henry's law, it's just how much gas can then dissolve and form carbonic acid, and the carbonic acid then drives the dissolution of calcite because it's just a buffering, right? So theoretically then, if you measure pH and you know the total alkalinity, you can calculate the partial pressure of CO2. So I won't get into that too much, but you can go and read about it. A good reference I can suggest is a book of Tony of Apollo and Postma. It's like the geochemistry Bible for me, but really good book. Anyway, so what you see here is the clustered data modeled in PHREEQC. So, and then this black line is just my little batch experiment to see if it could work. So then we theorized, okay, DOC can probably explain most of the pH behavior that we're seeing in the system, and you can and the model also effectively or really effectively predicts the relation between gypsum and sulfate. So what we did then, we said, okay, well, obviously the gypsum and the calcite and so on is you can't just estimate the parameter. We could really get really good normal distributions from the hierarchical clustering, because in fact, what our hierarchical clustering does, it normalizes data because it finds samples that are more or less the same, right? So you will have normal distributions. So it's a valid argument. Well, at least it seems to be in this case. So if you also then sort of assume a normal distribution for the acetate that gets input at various depths, and you just run a sequential reaction model, forward model, but it's sort of it's more sensitivity analysis. So you basically take every time a point that has the highest pH and you titrate it, or you add acid, right? We could more or less explain what is happening in terms of the pH CO2 dynamics. So this gave us a good, it showed really positive results in terms of a Monte Carlo simulation, which worked really well to predict the dynamics of sulfate, calcium, and magnesium in the system. So how to go about it? Well, I'll just quickly show you just inside of PHREEQC for the guys that do know. When you have equilibrium phases and you constrain the system, you tell the system, okay, you want calcite, dolomite, and gypsum to react until it reaches equilibrium. But then you can also define the amount that you add to batch experiment. So in this case, if you make it 10, there's ample and it will reach equilibrium. But if you make it sufficiently low, it will never reach equilibrium, but it will try to, right? So it will use up all this moles that you define for it, but then it will try to get to equilibrium, but obviously will not reach it because there's not enough reactants when you add DOC. Okay, so you have to define DOC. It's not in the default PHREEQC.dat database. Perhaps it is, I can't remember, but it doesn't matter. You can just define the kinetics or the thermodynamic parameters yourself, or there might be some other databases that might contain it. But you can do the same by just varying the PCO2, so the partial pressure of CO2 under equilibrium phases. So anyway, so this model more or less worked well, but the problem is that did not predict the sulfate values really well, because in effect what it does is it takes a point here and it reacts it sequentially. So you had this sort of weird lines going all over the place. But what we could do then is we could do away with the sequential modeling and just vary the PCO2, because we know this is related to the acetate or DOCO2 oxidation. So if you then have a normal distribution also for the PCO2, and you've also the distributions for gypsum, dolomite, and calcite, you can run a Monte Carlo simulation. And this I did in PHREEQC, but with R, and I will show you in a moment how you can set this up. But running 10,000 models actually predicted the behavior really well. So this is the pH versus PCO2. As you can see, it's relatively accurate. I haven't calculated the errors of the model yet. This is what I'm busy with now. It's not it's not always that easy to define your confidence in the model with Monte Carlo, especially if you have a lot of parameters. But for now, it looks really promising. And then also calcium versus calcite. It's just it's really fairly nice results. As you can see, the gray light dots in the background is my models, and obviously the colored dots in front is the clusters we identified in the system. And then in terms of sulfate, so this is where it gets a bit interesting. So we could predict whether sulfate is coming from riparian zone. So the model could not effectively predict the concentrations, but if you just run a simple evaporative model, so you take the shallow flow line, you can take the mean, or you can take the centroid of the cluster, and you just evaporate water. It sort of more or less effectively explains the behavior that we see in terms of sulfate. And then again, this is the gray dots on my model, and the other colors, the dots are the measured values. So effectively, what we could prove then is pyrite to explain the behavior of sulfate in the system. It seems to be all coming from secondary minerals, so gypsum or anhydrite. So you can more or less model those whole system like this, and there's a substantial amount of buffering in terms of calcite, but also if it's dolomite of magnesium calcite, this we have to now investigate through using calcium isotopes. And then also in the future we'll be doing some sulfate isotopes to distinguish between what is actually coming up with how the models predict. And it's a fairly simplistic model, just testing a hypothesis. So I wanted to show this today because it's quite interesting, and you can do most of this stuff yourself. And I'll show you where to get it. So for you, the guys that's working in R, which I would now recommend, I also model in Python, but it's a bit more difficult to PHREEQC because you have to create your own DLL, dynamically linked library. You can read about it, just install the library, and you can obviously define your input file. So
while I think they moved over, let me ask a question. So as a typical South African, I see you've got nearly 4,000 samples. Now, my question would be, do you think you would have obtained the same results if you only had 400 to work with? Because we'll never have the luxury of running a project with that huge amount of samples in the database.
Yeah, so one of the things, so I just have to also say that it was quite a big mission. So initially, when I started, I only had data for 3 borals. A lot of the data, a lot of the samples was still in storage, and we appointed a lab technician to run the samples. So in 3 years, he did 5,000 analysis. So I didn't have the data at the time. So what I did is you can also then create synthetic data, right? So if you make an assumption about the distribution of your data, you can run, you can generate your own data, right, to test it. Obviously, then statistically, you have to be really confident in your statistics, like if the mean is really the mean and the standard deviation. And I understand a lot of the times you will have weird distributions of your data, but it's doable. So you can effectively create your own data to test models. So, but again, this goes a bit more into the domain of multivariate statistics, and yeah, sometimes you should be a bit careful with that, but it's definitely an option, because I did this in the beginning to just test if it could work, you know, just a rapid prototyping to see if this type of modeling will in fact show us what we want to know.
Yeah, clever approach. Tell me, Nicolaus, speaking about the sampling itself, do you guys use any specialized sampling equipment? Do you preserve the samples, or was it just a normal grab sample at depth and that type of thing?
No, actually, this experimental catchment is really well instrumented. So all of the borals are sealed and have a it has a cup at the bottom, and obviously a perforated screen. And we use a peristaltic pump, and then they have the automatic data logger. So there's not a lot of water in this catchment. So in winter, for instance, or in summer, a boral might give you 1 liter of water in total. So you will pump it dry anyway, or if you pump it really slowly, you might have some recharge of the boral. So for the year, I did almost 2 years of my own sampling. For this, I used a inline sampling sort of mini lab. So it had 4 probes. It had a dissolved oxygen, EC, redox, and pH. And so then as you sample, you can measure these parameters and see when they sort of stabilize, and then you can take your sample. But for the most cases, for the last 10 years, it was just a bit of purging the boral until the EC stabilized, take the samples, and then they come directly to the lab where we filter them in 45 micron and acidify the ICP samples, and then the other samples go to the automatic sampler almost immediately. So the procedure in terms of the lab is really well sort of streamlined, and our labs are very well equipped to handle large amounts of samples, because on average, I guess, we generated about, just in the year I sampled, it was around 790 samples that we did.
That's quite a bit. Maybe just another question from my side. I don't see anything in the chat box yet, and I'll give some opportunity for other people as well. So I really like your results where you show the hydrograph, and you can clearly see the response of the clustering as one would expect. Have you gone to the stage yet where you actually look at let's take sulfate as an example. So you're measuring sulfate at that particular flow, and then what the model predicts, and how does that compare?
Yeah, so this is the sort of the capstone of my PhD project, and what I'm busy with now. We did 1 flood event that we had 15-minute incremental samples. So we processed about, it was a 6-day flood event. So I don't know how many thousands of samples those were. But obviously we didn't analyze each sample. We looked on the hydrograph and just sort of selected in areas where you have the change in concentration. And for this, we have major cations, anions, and stable isotopes. So this is now the last part, is so we'll do hydrograph separation with stable isotopes and then compare to what my model is doing to see how effective it is.
Any questions from the floor for Nicolaus? I've got one here. Given the importance of alkalinity in your modeling, how confident are you that the results, particularly for groundwater, are representative of subsurface conditions?
So you have to understand in the carbonic systems what alkalinity is. I can maybe just show you. So TIC is total inorganic carbon, right? But this is in fact what you're measuring when you're measuring total alkalinity. So what total alkalinity is a combination of various carbon species, but some of them do not exist in certain pH ranges. So if, for in fact, in most cases in aquifer systems, you measure total alkalinity and you have the pH, you can define the concentrations of your other species. So in our case, we actually measured total alkalinity, and we measured pH both in situ and in the lab. So we're fairly confident that we could define all the other species in terms of using then the relation of what the total or what total alkalinity is. I don't know if that answers your question completely.
Yes. Any more questions from the floor? Well, if you guys think of something, you're welcome to post in the chat. And if after our webinar, if there's some more questions, we can always get them to Nicolaus to answer. Nicolaus, we want to thank you for the time. I'm looking at my clock here. We're almost going for our load shedding, something that you guys in Europe are not used to. And so we plan our days around that. And yes, we wish you all the best of luck completing your PhD. And then, yeah, I've heard a little birdie sing. It seems like you're not coming back to South Africa, looking at other opportunities abroad. And that's actually a shame, because, you know, we value guys with your expertise in South Africa. But nevertheless, it's always be home for you, and the best of luck. Thank you a lot.
Thank you so much, Renier, and I appreciate all of you guys listening in. And this opportunity is really fun showing some results. I appreciate that a lot.
Thanks, Nicolaus. And so, hi to your wife.
I will do so. Have a good afternoon.
You too.
Bye.
Bye, everyone.